Another video on second PUC biology practical examination. In today's video, we'll be discussing the model question paper with the answers. Those who are new to my channel, please do subscribe my channel so that you don't miss any further updates on the board examination as well as competitive examination. Without delay, let's get started. So this is a model question paper for your examination of biology practical. So the first question will be a lottery system. Okay, lottery system, you have to pick up a chit and either you will get the question on the pollen germination from the given material and calculate the percentage of pollen germination. So for this is 1A. So 1A is 5 marks. Then either you can get this or show the pollen tube growth on the stigma and draw the label diagram of your observation. This is also for the 1A. Okay. Or you can get a question on TS of ovary of the given material and mention the number of locules and type of placentation, and you can either get nuclear staining and to show the shape of the cell and number of nuclei. Out of 4, you will get any one in lottery system. And the second question 2B is compulsory, that is regarding the mitosis. Next, in the third 3C1 and C2, 2 samples will be there. This is for your pH and it is awarded 3 mark. And in the 4D, you will be having the identification, 5th also will be on the specimen and 6th will be on the commenting on the F, and you will have a viva voce and the practical record. Viva voce is 4 marks, that is 2 questions internals will be asked and 2 questions externals will be asking. Okay, and practical, complete practical record you will be getting 6 marks. Okay, so this is a model question paper. So now I'll be discussing the key answers for it. Okay, if you get a pollen germination, the slide preparation you will be having 3 marks. I hope it's visible. Slide preparation. So what you prepare the slide, you will be having 3 marks, and for the calculation you will be having 2 marks. In the previous video, I have shown you how to divide the answer sheet to completely write your practical examination. So those who are not watched yet, please watch that video. I have given the link in the description below. Okay, so the pollen germination, so you have to write the tabular column this. So if you are taking 3 values, so this is hypothetical value, not this only. So in the microscopic field, so this is your microscopic field. So how many number of pollen grains you saw? That is 74, and out of that only 24 was germinated. So if you see the pollen grains like this, so this is the normal pollen grain, the one which is a germinated one, this one, pollen germinated. So you have to count the one which is germinated. So out of 74 only 24 was there. So you will get the percentage of pollen, that is n by n into 100. You know the formula for it. You have to mention the formula, that is percentage of pollen germination is equal to small n into 100 by capital N, that is 24 divided by 74 into 100. So this is one value, and if you are taking the 3 readings or 2 readings, you have to take the average. So the second reading you will get in the microscopic field, you will get be 78 and 76. Out of that you have to take the average. Here you will get the average, and here also you will get the average, what 24, 31, 28 divided by 3. That is how you take the average. Sum of all the 3 trials divided by the number of trials, that gives you an average. Okay, so then you have to write. Then the result you have to, below the tabular column, reference value is not result. Okay, so this is not a result. The percentage of pollen germinated is 36.33%. That reference line you have to write, and how much you have to write, what you are supposed to write, I have told you in my previous video. Okay, so if you get the question pollen tube growth on stigma for the question number A in the lottery system, so for the slide preparation you will be having 3 marks, and the label diagram you will be having 2 marks. So total it is 5 marks. So if you look at the diagram, you will be seeing the stigma, the pollen germination, pollen grain, and germinating pollen showing the pollen tube, and this is the stylar region of the stigma. So this is the microscopic view. You can see how the pollen is mounted on the stigma. Okay, so you will be having your stigma like this here, and upon it you will be having these germination. Okay, so these pollen grains appears blue color because of the stain, cotton blue stain. I hope it is clear for this question second. So if you get the TS of ovary, so the TS of ovary, for the slide preparation you have to show the 3 marks. Whatever the slide you prepare and show, that is for 3 marks, and the label diagram 2 marks. And here mainly you have to mention the result. So if you write, this is a locule, this is a ovule, and this is a placenta. This is the easiest experiment, and for this you have to focus in the dissection microscope. Okay, so these two you will be handling with the compound microscope. I hope it is clear. The number of locules you have to show 1, 2, 3, 4, 5. Number of ovule, it is 10, that is 2 per locule. Type of placentation, it is axial placentation. Suppose if you get the question nuclear staining, for preparing the slide you will be having 3 marks, and labeled diagram you will be having 2 marks. Okay. So this is a label diagram. The cells are rectangular, and in each cell you will be showing one nucleus. So this is a microscopic view. You can see the onion cell here. This is a 1 nucleus per cell. Okay. The result what you are going to write is, the shape of the cell is rectangular. Number of nuclei in each cell is 1. Suppose this is all for the 1A. Out of 4 practical you will be getting any 1. Okay. Now the question number 2. So this is a mitosis, and it is a compulsory experimental question where you have to perform. For the slide preparation you will get 3 marks, and for identifying any one stage, I have told you PMAT, that is either it is a prophase, metaphase, anaphase, and or telophase. Don't write multiple phases. You have to identify any one phase. Either it can be prophase, or it can be metaphase, it can be anaphase, or it can be telophase. Okay. So you have to look for the particular key features of each of the phases, and you have to write any of the diagram. Either you have to write this prophase, you have to write the stage is identified as prophase, then you have to write the diagram, or it is a metaphase, or it's anaphase, or it's a telophase. So this is a microscopic view you can see. So the prophase, the cells will be like this. Okay. And in the metaphase you can see, if this is one cell, you can see the chromosomes aligned at the center. Okay. And in the anaphase you can see like this towards the pole, and in the telophase the 2 cells like this. So this is a type of identification you can do, prophase. So like prophase, if the 2 cells are so attached together, that is a telophase. The chromosomes at the equatorial plane, this is a metaphase, and the chromatids towards the poles, that is anaphase. Okay. I hope it is clear, students. So these are the key features. You can pause the video, you can take a screenshot and refer it. So prophase, the chromatin becomes condensed thread-like structures to form chromosome, and all the key features what I told you. So you just go through it. And the next third question is testing the pH for soil and water sample. So you have to mention sample C1, C2 tabular column. If pH is 6, it is acidic. If pH is 9, it is basic. So you have to write the nature of the sample. So next question number 4D. So the 4D, you can either get a TS of testes, TS of ovary, VS of blastula, or female gametophyte. So here also you have to just write 1 to 2 identification. You just identify the slide if you get the very big 3 circles. So it is TS of testes. Okay. Then TS of ovary, you can see the Graafian follicle and there are corpus luteum. So many of the things, you just see the diagram and you have to identify. For this you have either the pictograph is kept or the slide can be kept for the particular question. Okay, so go through the points. I have given 2 to 4 points for each of the identification. Key feature, whichever key is the sense what, that is a one characteristic feature that makes you to identify, yes, this is a slide. Okay, so pause the video and you can take a screenshot and go through it. Any of the 4 valid reasons you have to identify. Then question number 5E. So this is a disease causing organism. So if you get this is a Entamoeba histolytica, so it causes amoebiasis. So which is a disease caused, only things that you must be writing. So then if it is a Plasmodium vivax, it causes a malaria fever in humans. So this is important and you have to write the scientific name. So see, in the printed it is in italics, and when you are writing you have to underline separately. Then it can be kept this as a slide. Entamoeba histolytica is a slide, Plasmodium vivax also you can get a slide or a picture graph in college, in your college which they have shown, please do remember. Then in the ascaris, either the picture is kept, most of the time the specimen bottle is kept. Okay, so it causes the ascariasis. And the fourth one, Trichophyton rubrum, it causes the ringworm. It is a fungal skin infection. Only these things you have to remember and you have to write. I have explained you in detail how much you have to pay attention for this practical in my previous video. Please go through it if you have missed it. Then the 6F. In the 6F, you will get on the xeric plants. Either you will get the xeric plants Opuntia, Acacia or the Bryophyllum. So read the key points which I have given, take a screenshot, and these are the key points by which you can identify this as a particular plant, why it is a xeric. So mentioning the word xeric plant is important, whether it is a adaptation for xeric condition or a hydric condition is very necessary. So next is a hydric plants. So Nelumbo, this is a lotus, you can see. Why it is that petiole is long, then leaves shapes are wide. So these are the points I have given, please go through it. Then Hydrilla eichhornia. So you have to identify the particular plant and you have to write the key points. Okay. Then xeric animal. So this is a camel, everyone will identify. Just identifying the camel doesn't fetch you marks, you have to mention it is a xeric adaptation in animals. Here it's not a elementary class questions, isn't it? Just writing a camel doesn't fetch you marks. You have to write xeric adaptation in camel, xeric adaptation in kangaroo rat, xeric adaptation in squirrel. Only then you will get a mark, students. Please make a note of it. And you just go through the points what I have given here, or you in your practical manual. These are the points is there. I am not going in detail here. I am just telling you how to write the answers and how much you have to write. Okay. So this is regarding the xeric animal. And then for the hydric animal, either it is a prawn, fish or frog. Most of the time this is the excluded, but be prepared for these questions as well. Okay. So this is a hydric adaptation in case of animal. Then homologous organs. So this is tendrils of passion flower and thorns of pomegranate. Why it is homologous? First you have to be clear with the definition of the homologous organs. Then scale leaves of onion and spines of Opuntia, limbs of human, cheetah, whale and bat. Please take a screenshot of it and you can remember 3 points for each of the experiment that is given here. Next, analogous organs. Once again you have to remember what is meant by analogous organs. Definition. So it can be kept thorns of pomegranate, spines of opuntia, rhizome of ginger, root of carrot, wings of insects, wings of bird, mandible of cockroach and lower jaw of robin. So you see the pictures. So for each of the things you have to know the key points. Okay, if you read these 3 points for each of the diagrams or particular things, definitely it will fetch you marks. I hope this video is helpful for you guys. Go through this video in detail. If you study this much, definitely nobody can stop you by getting 30 on 30 in your biology practical examination. Thank you for watching the video till the end. Those who are new to my channel, please do subscribe my channel so that it will give me more motivation, and it's free guys. Definitely don't miss any information on the board examination as well as in your competitive examination. Share this content with your friends so that it will be beneficial for them, and subscribe those who are new to my channel, and like this content if my work is reaching you.